Hello there, ninth graders. This is Teacher Krishna, and welcome to our biology class for week number 14. One more time, thank you a lot for all of the effort that you have done into accessing the online lessons, and of course for completing all of the exercises that we have prepared for you. Just like always, before we start, log into Agilex, enter to the Google Classroom, download your document, and save it in the biology folder as it is specified right below. As we move, remember to take notes on your Word document. That is something very important that it will help you out to keep track of all of the important information from this video and PowerPoint presentation. It's always important to start with our end in mind, and this week we're going to study topic 3.01. And the end in mind for this topic is describe the stages of the cell cycle by explaining the process of mitosis and its role in the formation of new cells, so that he and she can describe the structure of a chromosome. So this might look a little bit extent, but do not worry. We're going to expand in each one of the key terms from here in a while. So we need to start talking about the cell division. And I have a question for you. How is it possible that a puppy doubles its size in 4 weeks? And this is very interesting to analyze because, I'm pretty sure that just like me, you have had pets before. And you have noticed that when they are just little puppies, after a month, they are huge, but literally they duplicate their size in just a very few time. So how is it possible that they can grow so fast? Well, the answer is found in cell division. It duplicates in the amount of body cells that it has. So here's an example. A little puppy, and 4 months later, now we can see that it looks huge. So you need to know that the cells that we have in our body are constantly going through what we call cell division. And that is the creation of new cells. But especially in puppies, it's very noticeable because they duplicate all of the cells that they have in their bodies. And at the end, the result is basically having a bigger puppy. And here is what all of the steps and stages of the cell division look like. We have G1, which is the first phase, and it is basically cell growth. We have S phase, which is the DNA replication. G2 phase, that is the preparation for mitosis. And these 3 steps are part of something that we call interphase. And lastly, we have another division that is cell division, which is the M phase, which includes mitosis and cytokinesis. So we're going to expand a little bit more on each one of them. Let's start with the interphase. So in order to successfully complete all of the steps in the interphase, it will take most of the cells around 20 hours. The first stage is G1. So basically, during all of this section, cells will have a normal function. And the cell grows in size until it cannot continue with its normal functions. So let's suppose that we're talking about one of your blood cells, for example. So basically, cells are going to keep on growing a little bit until they can divide, but they will keep on working normally. We have the S phase, copy of the chromatin and DNA information. Basically, what happens in the S phase is that we are going to duplicate all of the genetical information that we find in the cell. Because when they create a daughter cell, then both cells need to have the same genetical information. And how is it possible that they get the same? By duplicating it before they divide. And then we have a G2, and the cell grows a little more, and it is ready for the cell division. And one more time, here are the 3 steps of the interphase. We have G1 or G1 phase, we have S phase, and G2 phase. Now we're going to continue with the cell division. So mitosis. The cell distributes the genetic material created in the S phase. Exactly. So you're going to find later on that mitosis has several steps to follow. But basically what happens in all of them is that we are going to divide the genetic information in them. And we have the cytokinesis as well. Two identical cells are created at the end, and they are named daughter cells. And of course, they are ready to repeat the whole cycle again. So, before we continue, what interesting facts can you find in the Did You Know section? Okay, that is in page number 2 of the topic we're studying this week. So I'm going to give you a little bit of time to go to page number 2, find the Did You Know section, and of course find some interesting facts from there. Just like you can see here, in the cell division we have the M phase, which include both of the things I have mentioned already, the mitosis and the cytokinesis. And even though it might seem like the smallest process that the cell goes through, then it is a little bit more complicated than the previous phases. Now we're going to study them right now. We have DNA and chromosomes. So DNA is a genetic blueprint for cells. So this is something very important to analyze. Right now, each one of you has different characteristics, like if you are tall, if you're short, if you're skinny, or a little bit more chubby. But all of that is determined by the DNA and genetic information that you have in your cells. Even the color of your eyes is determined by your DNA. Right? And most prokaryotic cells only contain a single circular DNA. So, remember that prokaryotic cells do not have a clear nucleus. So, that is why they only have one circular DNA, which is different in eukaryotic cells. Remember, eukaryotic cells are found in animals and plants. And eukaryotic cells store DNA inside the nucleus. And proteins wrap up around nucleosomes. So later on you will start studying how DNA forms, but basically proteins take a huge factor into the creation of DNA. And here we have a small pattern that cells, or most likely that we need to follow in order to create a chromosome. So first of all, we have the DNA information. So once a lot of DNA gets together, they assemble and they create a gene. And after DNA creates a gene, and after genes get together and assemble together, they create a chromosome. But they are all, one more time, all of the genetic information that basically gives your body all of the specifications that it has. And something interesting is that humans have 46 chromosomes in total inside their cells. Okay, there is one exception to this, and the only cells from the human body that do not contain the 46 chromosomes, but that have 23 chromosomes, are the sex cells, which are the sperm and the ova. But don't worry, we're going to study that later on when we talk about the meiosis process. Here we have a picture of what DNA looks like. And this is something interesting because it is made by a double helix, and inside we have several nitrogen bases that pair up in very specific and clear patterns. So if they don't pair up in like a normal pattern, we can call it like that, what is going to happen is that a mutation is going to be generated in our bodies. Right, we have the mitosis process. So, process that copies the chromosomes into different nuclei. Their stages are. So, the first one is the prophase. So, in the prophase, the chromatin condenses into chromosomes. They look like a letter X. The nucleus starts to break down and spindle fibers attach to the centrioles. So this might look a little bit difficult to understand, but don't worry, I'm going to show you a picture and it's going to be clearer later on. We also have the metaphase. The chromatid lines up in the middle of the cell, and spindle fibers attached to the centromeres and the chromatid. I'm sorry, that should be as a letter C in there. All right, so here we have the prophase. So you can see those like orange X's that we have right there. Those are the chromosomes. So basically what happens is that the DNA information, when the cell is not going through reproduction or through the cell cycle, then the DNA looks just like spaghetti and it is just spread around the nucleus. But when it starts during the prophase, what happens is that the chromatin starts getting together in very specific forms, just like the one that we have right here that are like X's. And you can notice that around the yellow circle, the greenish lines that we have right there are not complete. So what happens is that the nucleus is starting to break down because later on all of that genetical information in chromosomes are going to go out of the nucleus, okay? Now here we have what happens in the metaphase. So now here we cannot see any other nucleus because it already disappeared. But what happens right now is that the chromosomes were originally in the nucleus are right in the middle. And just like you can notice, in both ends of the cell, there are like this orange teeth, and those are called the centrioles. And you can also see several blue lines. Those blue lines are the spindle fibers. And they are very important because this process is very organized. So we have centrioles in both ends and we have spindles attached to the chromosomes, and lastly the chromosomes are aligned right in the middle of the cell. Those are the first two stages of the mitosis. Let's continue. So we have the anaphase and chromosomes are pulled apart. The individual chromosomes move along the spindle fibers to opposite ends of the cell. We have the telophase. The chromosomes form the chromatin again, and the spindle fibers break down and the nucleus is formed. Okay, so first of all, this one is the anaphase. And just as you can notice, now the chromosomes don't have the X shape anymore, but now they're just like a C or an inverted C. So what happens right now is that the genetical information is being spread apart. Because just like I told you before, at the end, both cells are going to be identical. They are going to have the same amount and the same DNA information. Okay, so basically in the anaphase, what happens is that the chromosomes separate in order to divide the specific genetical information. And now, this is something interesting. This is a telophase. And you can now see the nucleus inside the cells. The nucleus are those yellowish circles with orange particles and the green edges. Because in the telophase, what happens is that the genetical information gets together one more time and it assembles inside the nucleus. And just like you can notice one more time, it is not like clearly divided or compressed as in the chromosomes, as when they were X's, but now it is just spread around. So usually this is the way the nucleus looks like and when the cells are in a normal stage, because they are not, the DNA information is not condensed, it's just simply spread around. And you cannot see any more blue lines around, you cannot see any more spindle fibers because they break down, because they are not necessary anymore, because the division has been completed. And then we have the cytokinesis. It finishes the process by splitting the cells in two. It divides the cytoplasm and rest of organelles in equal parts. So the cytokinesis is the last stage of the cell cycle. And just like it was specified in here, the cytoplasm is divided in equal parts as well as the organelles. This process differs in animals and in plants, and this is something very, very interesting. So, in animal cells, the cell membrane is pushed inwards, and in plant cells, plates form in both ends of the cell and they separate. So here's an example. So in plant cells, what happens is that they have a cell wall. So remember that plant cells are a little bit more rigid because they have the cell wall and they have a cell membrane. So the membrane cannot be pushed inwards. So basically, just like you can see in the left, they form some plates around and they simply separate. But on the right, we have the animal cell. And what happens right there is that the membrane starts compressing inwards until they both separate. So I really like the videos that we can find in the platform. So before we continue with everything, go to your topic on Agilex and watch the videos. They are great to look. Okay. Right, now let's go to our evaluation. So students have to read and summarize the content from topic 3.01, especially page number 4. So I already summarized most of the content that you have in the platform, but it's always good that you go one more time and reread it. So remember that you are going to learn things better if you take a look at them once, twice, and 3 times even better. So when you are done, you have to complete the lab report on page number 5 of the same topic. So you will find a virtual lab in there, and it's very important that you complete all of the aspects that are specified. Please take your time to read all of the aspects that you find in your lab report. And if you have any questions, you can contact me in the Google Classroom, or you can ask me any questions that you might have during the Zoom session. And this week, it's important that you participate in the discussion during the Zoom session. I will be assigning some points as well when you participate in the Zoom session. Okay? So that'll be it for today's class. Goodbye, guys. I will see you next week.